What's up guys, we are back. In today's video we're going to continue talking about the biology section from the IMAT 2025. So if you haven't watched the first video, I strongly recommend you to go there and take a look before we move on in this one here. So let's go straight to the point. I have the IMAT paper open here on my computer, and you know what, I just realized that in the last video I skipped question 13. So let's start with this one and then we work ourselves through the next one. So question 13, it's kind of following the pattern of the previous questions we saw in the previous video, that is all about cell respiration. So let's take a look at this. So 13, oxidative phosphorylation is a metabolic pathway, 2 dots. So it wants us to describe what it is, okay? And we gotta try to find what is the best description. Now, once again, I mentioned this already. The purpose of this video is just for us to analyze the questions and try to figure out why option A, for example, is the correct one and why the other ones are incorrect. So we can try to learn a little bit from it, because it's what I always say. The questions from the biology sections are mostly very similar from year to year. They don't have much place to go, so they're always questioning the same thing in a different way. So if we could try to project what other questions they could make about this topic, we're gonna be way ahead. So let's go straight to the point. Letter A is saying through which the energy stored in reduced coenzyme is used for the synthesis of ATP. So the key words here that we have to pay attention is coenzymes, reduced, and synthesis of ATP. All right, so it's all aligned with what oxidative phosphorylation is. So if you have a clear idea about the cell respiration process, you're gonna remember that we're gonna have the glycolysis taking place in the cytosol, then we're gonna have the link reaction happening in the mitochondria, and then we're gonna have the Krebs cycle, and then finally, we're going to have the oxidative phosphorylation. That is that process that will give us a big, large amount of ATP. So remember, the oxidative phosphorylation is going to be taking place in the inner membrane of the mitochondria, and we're going to have 4 important complexes, which is going to be responsible for pumping hydrogen out on that intermembrane space, so we can create that gradient full of hydrogen that will later be used for the ATP production. Just remember, coenzymes, we're talking about NADH, we're talking about FADH2. These are important coenzymes. They're called this way because they're going to help the main enzymes. In this case, the complex 1, 2, 3, 4, they are actually the enzyme, and they're going to be carrying the electrons so the whole process can happen smoothly. Now, before we move on and take a look at the incorrect options and why is it wrong, I just want to mention that oxidative phosphorylation, yes, it's a strong topic for the IMAT. They might be asking again in a different way, but there's a different type of phosphorylation that happens as well that produces ATP that is not called oxidative phosphorylation, but it's called a substrate phosphorylation. This happens in the glycolysis. Actually, this is why we have a net gain of 2 ATP being produced, because we're going to have a phosphorylation that happens in the level of the substrate. So this might be a question that the IMAT could probably be making, you know, like trying to know if you understand what is the substrate phosphorylation and where does it take place. Keep that in mind, put it in your notes to take a look later. I think that is very valid for you to be aware of. Now, letter B here is saying through which the amino groups resulting from the degradation of amino acids is eliminated. So we're not eliminating nitrogens, we're not breaking them down, okay? It's all about producing energy, not like cleaning up the toilet after a protein shake. This is basically the urea cycle, so we're talking about different things. Letter C and letter E, they're kind of obsessed with fatty acids. Letter C it's all about breaking down these fatty acids 2 by 2, and letter E, it's actually talking about synthesizing the fatty acids, okay? So the letter C, it's a very interesting one, because even knowing that it's not the correct answer, it's talking about beta oxidation, okay? And beta oxidation, it's a topic that the IMAT could be talking about, okay? So how can we produce energy through fatty acids? So the beta oxidation also happens in the mitochondria, in the matrix of the mitochondria, but it has nothing to do with the oxidative phosphorylation. So we can eliminate letter C and letter E. Finally, we have letter D here saying that which represents the final stage of the anabolism of amino acids, carbohydrates, and fatty acids. So it's one of those questions that wants to seem a little bit smart using those like hard words, you know, but it's really, you can tell that there's something off, you know. It's almost almost like a fruit salad here. It's talking about that oxidative phosphorylation. It's going to be responsible for the final stage of anabolism of amino acids, carbohydrates, fatty acids. Guys, when it comes up to biology and biochemistry, like everything is really organized and everything has a name and everything has an enzyme. So you're not gonna see any process that actually happens like a unique process that converge different types of substrates for the same purpose. So you can kind of run away from a sentence like this. So it's completely incorrect. This leads us to letter A, obviously. I hope this explanation was clear. Let's move on to the next one. All right, guys, so our next question here will be the question 15. Just remembering you all that question 14 was covered in our previous video. So we're gonna jump here to the question that talks about the collagen, all about collagen, the single word that is keeping a whole multi-billionaire industry of skincare alive. So basically this question just goes straight to the point. So collagen is a protein that is, and then the options. So extracellular was the correct option. Then we had membranous, intracellular, abundant in a thick filament, nuclear. Like, I don't like questions like this. It's like too wide open, you know, and sometimes if you start thinking too hard, you might let your brain lead you to places you shouldn't go. But whenever you find a question like this and you encounter this type of open, wide question, you go straight to the most obvious one you can find. In this case here, it's the extracellular one. Okay, so you guys remember that the collagen, it's all about keeping the cells well structured. It's almost like the cytoskeleton of the cell, but from the outside. It is a huge protein, really big. This is also one of the reasons why some people say that applying some products with collagen in it in your skincare doesn't really work because your body cannot absorb that, but who knows. Now, the other options we had here, like letter B, for example, is membranous. You might think that, oh, okay, it's flexible in a way, and it is in a way if you think about it. The collagen is so important even for the bones. If you have any kind of problem in producing collagen, you can develop a disease that is called the brittle bone disease. There's actually a really cool movie you can watch. It's called Unbreakable, and the main character has this type of disease where his bones are so fragile that any type of impact, it's enough to break them down. But in this case here, the word membranous, it's actually screaming membrane cell. Okay, so those proteins that we have, that sometimes they formed some channels to let some ions go inside out. So we have those integral proteins or the peripheral proteins that is usually like responsible for some signaling. This has nothing to do with collagen. That's why this was not the correct option. Letter C, it's obvious for us. It's basically the opposite of the correct answer. Letter D, it's talking about filaments, so the contraction of muscles. Is trying to make you think that might be related with actin and myosin, which is not the case. And letter E, it's talking about the nuclear. And you guys know that if there's any structure situation taking place there, it's thanks to the cytoskeleton of the cell and not the collagen. So let's move on. All right, guys, so our next question here, question 16, it's all about the muscle contraction. Okay, so this is the first question of 3 questions that we had in this edition of the IMAT that talks about the muscular system. The IMAT loves covering topics of systems in general, so I really recommend you to keep up your study on that. Now, because we had like 3 questions in this edition, I'm not quite sure if we might see this in this year, but we never know. Okay, I have some like strong feelings that the IMAT might be asking some questions about the renal system. Okay, I think this is a very strong candidate, and also about the neural system. Giving us some attention, some extra attention to these 2 systems wouldn't do any harm. If you could, I would strongly recommend. Now, this question is saying that muscle contraction is triggered by an increase in the concentration of calcium ions that binds to. This question is basically like the classic love story between actin and myosin wanting to connect to each other, you know, in order to be able to promote the muscle contraction, but not being able to because there's someone blocking the way. So calcium will be like this wing guy that will guarantee that this could happen. Okay, so he's not going to bind to the main characters, so he's not going to bind to myosin or actin, and neither he will bind to that block rope, okay, that in this case is the tropomyosin. This protein in particular, it's going to be blocking the sites of actin where myosins usually connect to. So once we have calcium, this is a guy that's going to be moving the tropomyosin out of the way so this connection can happen. Now, about the options here, so you have myosin and actin, like naturally they will be there when talking about muscle contractions. This is the first thing that comes up in our head. So if you have like a basic understanding of muscle contraction, you might choose one of these 2 options. People don't usually tend to remember about these 2 characters here. Now, letter C is the ultimate trap of all these questions here. But you need to remember that who's going to be doing the hard work here. It's going to be the troponin, okay, that is present in the actin. And letter C, viamentin. Like, honestly, I don't even know what it's doing here. So let's go check the other questions. It's also about muscular system. All right, guys, so moving on here. Question 17, still talking about the muscular system, but we're gonna actually, it's a sequel, okay? We're gonna see the other side of the moon here. So let's take a look. Which of the following statements about myosin and actin is correct? Okay, do the brainstorm as always. Keep the question in mind, and let's go to the options. Letter A, the binding of ATP to the actin-myosin complex promotes the dissociation of the complex. Obviously, this is the correct answer, and it's basically saying that whenever ATP comes into play, okay, and binds to this complex, they are going to dissociate, okay? Once we just saw, for example, how does the muscle contractions happen, and calcium is super important for this to take place. And once the actin and the myosins are together, they don't want to be apart anymore, okay? So they're going to continue together as long as possible, and the only thing that can actually break them up is money, okay? Actually, biological money. So we're talking about ATP. So the ATP is going to be the one responsible for binding this complex and dissociating the complex. The sentence is perfect. Now, this question in particular here, in my opinion, I think it could be considered like a medium hard question, because it kind of forces you to think in a very particular way that ATP, it's going to be the one responsible for later on being broken down, and that energy will be storage, like a spring. That's going to be the energy that will make the myosin just go back. So the reaction can happen once again whenever we have that impulse of calcium. Let's take a look at the other options here. Letter B is saying that the binding of ATP to the actin-myosin complex promotes the formation of the complex. It's basically the opposite, the other way around, right? So if you would have been indecisive between the options, maybe it would be between letter A and B. The letter C is saying that the binding of calcium to troponin is necessary for the dissociation of the actin and myosin complex. We have already gone through this. We know what troponin is responsible for, and it's not for dissociation. Letter D is saying that actin has ATPase activity, and letter E is saying that tropomyosin, that is that protein string that is present in the actin, has ATPase activity, okay? So both of them are wrong. The only protein that actually has the ATPase capacity is going to be the myosin. So the head of the myosin is going to have this ability. So this is an example of how deep the IMAT can go into some topics. So the best thing for you right now is to go and find a list of high yield topics and really study yourself through every single details of these particular topics. All right, guys, so last question of this video here. We're going to continue this playlist, okay, with further videos until we finish all the biology questions. But let's take a look at question 18. So question 18 is talking about the Bohr effect. And guys, believe when I say it, this topic here is like one of the high yield topics. It shows very often in the IMAT. It shows an ear, sometimes it doesn't show in the other one, sometimes shows 2 ears in a row, okay, because there's a lot of things we can talk about this, and there's different ways we can ask about the Bohr effect, and we can correlate this topic with other topics of physiology in general. So it's really good for you to have a good understanding on this. I have already scheduled a video just to talk about the Bohr effect. Let me know if you guys are interested on this. So let's see. The Bohr effect is observed in tissues that are oxidizing large amounts of nutrients and generating hydrogen ions and releasing CO2 into the blood. This results in, first of all, so it's talking about the Bohr effect, and a key word here is oxidizing, okay? So it's saying that we see it very prominently whenever we have large amount of oxidation going on. And remember, we're talking about cellular respiration, beta oxidation, so everything that is breaking down, catabolism, okay? We're breaking things down, proteins, fatty acids, carbohydrates, okay? So there's a lot of oxidative phosphorylation going on. All of this will generate a high amount of CO2, okay, along with hydrogen ions. And what does hydrogen ions do? Remember that this will lower the pH of the environment, okay, of the tissue in particular. So let's think about tissue that produce a lot of CO2, produce a lot of metabolic stuff. The muscles, for example, we are always using our muscles for everything. So you can tell for sure that the Bohr effect is happening really strongly in this tissue. And if you take a look at the options here, we obviously know that the letter A is the correct one, okay? But if we look at each one of them, we're going to see that in order for us to be able to answer this, we really need to understand what is the Bohr effect, okay? So there's no other way around. And in very short lines, the Bohr effect, it's going to be this event that whenever we have a lower in the pH of a tissue, okay, and a higher amount of CO2, the hemoglobin, okay, so we're talking about the blood cells that are carrying oxygen throughout our body, they're going to immediately drop down that oxygen and they're going to bind to these hydrogen ions. The reason, well, they have much more affinity with these hydrogen ions, okay, than they'd have with the oxygen. And not only that, the high concentration of CO2 in these ions will promote these oxygen drop down. So these hemoglobins will be binding majorly with the hydrogens, but they will also be binding with the CO2. This actually, it's a very tricky one, okay, because the CO2, the way it is transported outside of our body, it's not mainly through the hemoglobin, okay? It's actually through a process, chemical process, where the CO2 will be interacting with the water surrounding, okay, and will create a weak acid, and later will be converted into bicarbonate by releasing one of these hydrogens of the molecular form. This hydrogen is the one that is usually carried by the hemoglobin. So this is a dense topic that deserves a lot of attention, and if you didn't really know about the bar effect concept, it would really be hard for you to answer this question. Alright guys, so I hope you guys enjoyed this part of the video. There's not much more I can say about this, only that I have planned a video just about Bohr effect, but I need to know what you guys think about it. And in the following days, I'll be releasing more videos with more questions about the IMAT 2025 until we cover all the biology section. I hope you liked, I hope you enjoyed, and if you did, don't forget to like the videos and subscribe to the channel if you're new. I'll be seeing you guys in the next one.